In this video, we will discuss about KRAS mutation role in the cancer cells. As KRAS mutations are found in most of the cancers, especially in solid tumor cancers such as pancreatic cancers, breast cancer, lung cancers. So it is important to know what are the main roles this KRAS mutation plays that helps in the immune suppressions or tumor progression. So this KRAS mutations, we will start with this cell surface markers or cell surface receptor changes. This KRAS mutation will cause high expression of the CD47, and this high expression of CD47 can help in the polarization of the macrophages from M1 to M2. And M1 type macrophages has this anti-inflammatory properties or tumor suppression properties, whereas M2 type macrophages has tumor proliferation properties, or it helps the tumors to grow. So high expression of the CD47 will help the polarizations of the macrophages from M1 to M2. And people have also found out that by inhibiting this CD47, they are able to see reductions in the tumor suppression. And another receptors that these KRAS mutations upregulate regularly in most of the cases is CD155. The CD155 upregulations can interact with this TIGIT on the T cells, and it will also helps in this pro-tumoral tumor-associated macrophages, or we can say that M2 type macrophages. So by inhibiting these two cell surface receptors on this KRAS, people have seen that there is reductions in these immune evasions, and they can revive the immune sensitivity against the tumors with KRAS mutations. Other things that this KRAS mutation does, that it take advantage of the high ICAM1 expressions, especially in the tumor microenvironment, by this M1 macrophages. And this high ICAM1 interacts with this KRAS, and they also then polarized this M1 type macrophages into the M2 type macrophages. So it takes advantage of this high ICAM1 expression of the macrophages in the tumor microenvironment. Other things, or other important thing that this KRAS does, is it changes the surrounding conditions. That is, using this HIF1 alpha, it creates a hypoxia conditions or low oxygen environment around this KRAS, especially in the tumor microenvironment. And this HIF1 alpha will cause release of this lactate and CSF2, and they will also promote this pro-tumoral tumor-associated macrophages. This tumor-associated macrophages is mainly the M2 type macrophages that helps in this tumor proliferations. The other important thing that it does is it reduces this IRF2 secretions. Reduction in the IRF2 secretions will cause the release of this CXCL3, and this release of this CXCL3 can interact with this myeloid-derived suppressor cells, and it helps in the recruitment of these myeloid-derived suppressor cells to this KRAS region, or in the tumor microenvironment near the KRAS mutated cells. And this myeloid-derived suppressor cells will also be polarized into this M2 derived macrophages. So the important thing that this KRAS mutation does, or one of the common things that find out in this figure, is that it tries to polarize these M2 type macrophages into the M2 type macrophages through different means, or through different pathways, such as this HIF alpha pathway by creating a oxygen deprived condition, or through the high surface expressions of the CD47 and CD155. And this effect is not only limited to the macrophages. Once the macrophages converted into the M2 type macrophages, then it will suppress the other immune cells such as T cells also. And we have seen the interactions between macrophages and T cells when we discussed about selectin interactions with pancreatic cancer cells. That is, pancreatic cancer cells integrates subunits, ITGA3 and ITGB1. There we have discussed, if these interactions are being suppressed, then macrophages will not convert it to the M2 type macrophages, and thereafter it will not inhibit the T cells, activations of the T cells. So the M2 type macrophages has other effects, has effects on to suppress the other immune cells also. So I hope you understood this main rule or some of the important functions of these KRAS mutations in the cancer cells. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.